Right, I think we've reached 1 o'clock in the UK, and it's my pleasure to welcome you to this final presentation in the series on water. It's also my pleasure to introduce Dr. Sarah Donoghue, who's our final speaker. Sarah's first degree was in environmental geology from the University of St Andrews, and she followed this with an MSc in GIS and a PhD in predictive modelling of soil-lead concentrations using GBase datasets for urban environments, both of these at the University of Edinburgh. GBase stands for Geochemical Baseline Survey of the Environment, and this is the National Strategic Geochemical Mapping Programme in the UK that was carried out by the British Geological Survey, who were also co-sponsors of Sarah's PhD project. So before her PhD, Sarah spent some time working for the Scottish Government, and since her PhD, she has taken on postdoctoral research projects sponsored by Scottish Water and also by the Scottish Environment Protection Agency, SEPA. And I'll let Sarah say more about these as part of her presentation today, which is titled Water Quality Monitoring, Current Procedures and Future Models.
Thank you very much, Margaret. So I'm just going to share my screen. Hopefully we can all see that. Let me know if we can't, because I can't see anyone. But yeah, thank you for the fantastic introduction. So yeah, I'm Dr. Sarah Donoghue at the University of Edinburgh. And as Margaret alluded to, my PhD was actually on modelling contaminants in soil. So I'm relatively new to the whole water world. But the work I'm going to talk to you about today is based on two postdoctoral projects that I've recently completed. So one for Scottish Water and the other for the Scottish Environmental Protection Agency, SEPA. So I'm not actually currently researching water quality as I'd like to be doing. This is due to a current lack of funding, but I'm currently pursuing different funding options and I'm looking for international partners. So please drop me an email if you think your research complements my own and would like to work with me in the future. So today I'm going to discuss six key questions around the central issue of water quality monitoring. So first of all, in case you're not already convinced, I'm going to hopefully convince you why it's important to monitor water quality. I'm then going to look at how climate change might have an effect on future quality of water. I'm briefly going to discuss some differences in how water quality is monitored around the world. This will lead into how water quality is monitored in Scotland. And then I'm going to discuss some of the results from a project I did for SEPA, looking at analysis of their water quality data set and lead this into future water quality monitoring procedures, perhaps using machine learning modelling. So first of all, you're probably all very aware of the negative impacts poor water quality can cause, but I want to spend just a few minutes summarising some of the main adverse effects of poor water quality, just so everyone's on the same page. So it goes without saying that the adverse health effects from poor water quality are well established worldwide. This sobering graphic on the left, I got made my mouse visible, shows the number of deaths related to unsafe drinking water, and it's still a massive issue in many countries. In fact, 1 million people are estimated to die every year from diarrhea as a result of unsafe drinking water. And although death is clearly a severe result of poor water quality, the adverse health impacts related to contaminated drinking water range from skin inflammation and nausea or vomiting to neurological degradation and some types of cancer. This has led to recommended maximum safe levels for numerous contaminants in drinking water. I've listed some of the most harmful contaminants in this table here, along with the recommended maximum guideline levels for several different governing bodies worldwide. And you'll notice there can be quite some variation. For example, manganese in this row, which continued consumption of high levels of manganese in drinking water can cause neurodegenerative disorders, cardiovascular diseases, and liver damage, had 60% higher guideline values under the WHO compared with the UK Environmental Agency and the US Environmental Agency here. So there's clearly a lack of standardization. You'll also notice that cyanobacterial biomass and microcystins, a cyanotoxin, are only recommended under the WHO, and these are very low levels. So these limits occur due to the health impacts caused by cyanotoxins produced by harmful algal blooms. There's an example shown here for Loch Leven in Scotland, which is quite close to me. Direct contact with cyanotoxins from these harmful algal blooms can result in asthma, skin irritation, and ingestion of these toxins may lead to gastrointestinal upset, muscle weakness, acute liver damage, neurological disorders, and very infrequently death. However, massive oxygen depletion may also occur as a result of harmful algal blooms, which may cause the death of many aquatic organisms, as demonstrated up here. Eutrophication is just one of the many adverse effects poor water quality can have on ecosystem health. Overall, polluted waters can alter food webs, weaken ecosystem resilience, and threaten species that depend on clean water for survival. Poor water quality can also cause economic impacts through increased healthcare costs, reducing agricultural productivity and the quality of the food grown, limiting tourism, and many more that are listed here. So this diagram is taken from a recent article by Russ et al., who looked at the impact that water quality can have on GDP. So they found that heavily polluted waters can cause a 2% decrease in downstream growth on some rivers, and this depends a lot on the economics of the country being studied. Therefore, monitoring or regular measuring of water quality is essential for protecting human health, preserving ecosystems, and supporting economic activities. Monitoring usually involves collecting field samples and observations, testing these in the labs, and then comparing them with recommended guideline values. So this involves looking at the physical, chemical, and biological characteristics of a water body. And this can vary both spatially and temporally, and this can affect the accuracy, which I'm going to come back to a bit later. But simply, what water quality monitoring wants to achieve is an early warning system so that we can know if and when adverse health effects are likely to occur. This means we can properly plan for these events, either by adapting our policies and management practices now, before it's too late, or finding ways to adapt for these changes in the future. It's also useful to have baseline measurements of water quality so that we can determine if a water body is degrading, either due to anthropogenic or natural processes. Therefore, we can assess the impact of future environmental trends, such as climate change. So this brings me nicely into my next topic, which is how climate change is likely to impact water quality worldwide. So a recent article by Van Vilt et al. In Nature summarized that intense hydroclimatic extremes linked to climate change, such as droughts, heat waves, rainstorms, and floods, are likely to deteriorate river water quality. This is driven by altering hydrological cycles, increasing water and soil temperatures, and the interactions between hydroclimatic, land use, and human drivers. For example, drought conditions, as seen in image A in Texas, can elevate salinity levels above their maximum threshold, which means they're unsuitable for use in crop irrigation, as illustrated in this graph. In image B, heatwave conditions in a European river caused cyanobacterial blooms, which I've just described, and these decreased oxygen levels below the ecological threshold, as illustrated by this dashed red line. In image 3, we see flooding in a Western European river, and this elevated lead concentrations above their safe guideline level, making drinking water from this river unsafe. So to explore this in a little bit more detail, I'm going to look at one part of the water system, the biogeochemical cycling of phosphorus. I generated this figure when I was reviewing how eutrophication, a sudden increase in nutrients, can lead to harmful algal blooms, which I showed images of earlier. And the occurrence of harmful algal blooms is dependent on phosphorus and other nutrients, including nitrogen, and the cycling of these in the reservoirs or lakes and the sediment. However, you'll notice that many parameters in the cycle are vulnerable to climate change. For example, inflow into the system from heavy rain events could increase the levels of suspended solids in the water, along with carrying more phosphorus and other pollutants from anthropogenic practices like fertilizer addition, sewage generation, and soil erosion. Secondly, an increase in air temperature from global warming will cause a comparable increase in water temperature. And as the optimum water temperature for cyanobacteria growth is 25 to 35 degrees Celsius, even a small increase in water temperature may facilitate algal blooms at different seasons or for longer time periods. And this temperature increase could also extend the geographical range that water bodies are at risk from cyanobacteria. Thirdly, stronger wind-driven currents are predicted to be a result of more intense storms due to climate change. In shallow lakes, this may cause the resuspension of lake sediments that may contain phosphorus and other contaminants stored within them and release these back up to the water. So lastly, the sediment-water interface is a really important part of the phosphorus cycle. Reservoir sediments tend to be a sink for phosphorus. However, when the external phosphorus load is reduced, for example, by different management or nutrient additions, the phosphorus stored in the sediments can be released back to the water and act as an internal source. So in the Northern Hemisphere, the highest phosphorus release from the sediment usually occurs in the summer. This is because the reservoir or water body tends to be thermally stratified in the summer, which you can see here. So the epilumnium, the upper part of the water body, has a higher temperature, and the hypolumnium, the water closer to the base of the water body, is colder, and this causes a temperature differential. As you can see from the diagram, it also causes a decrease in oxygen conditions lower down in the lake. And these anoxic conditions towards the sediment interface can cause the dissolution of iron oxyhydroxides and any phosphorus bound to the sediment. So during thermal stratification, phosphorus released from sediment is very slowly transferred back up to the surface. However, when the lake destratifies, which usually occurs in the Northern Hemisphere in autumn, there's a rapid increase of phosphorus back up to the sediment, back up from the sediment to the lake surface, which can cause algal blooms to grow. So when considering water quality, predicting when this sudden eutrophication of surface water is likely to occur is really important, which I'm going to come back to a little bit later. And the timing of this destratification can be impacted by climate change via the wind mixing and the temperature differences I just explained. So I'm now going to talk about differences in global monitoring of water quality, briefly look how it's monitored in different parts of the world. I'm sure some of you in the audience will know a lot more about this than me, so please correct me, maybe at the end, if I get anything wrong. To begin with, as the seminar series aims to discuss topics surrounding water security from Europe and Africa, I'm going to look briefly at South Africa. So in South Africa, there are national and regional water quality monitoring networks that have been operational since the early 1970s. So there's a wealth of data here. The Department of the Water and Sanitation in South Africa oversee the National Resource and Monitoring Programme. And this includes two key parts. So the National Monitoring of Chemicals, which looks at chemical pollutants in rivers, dams and groundwaters, and also the National Eutrophication Monitoring Programme, which aims to detect algal blooms in water bodies and track nutrient levels. As shown here, both these programs rely on data from remote sensing methods, also sampling, field measurements, along with laboratory analysis and field observations. This is used to detect contaminants and ensure compliance with national and international water standards, mainly WHO standards, but there are also South African water quality guidelines. All this data is combined into one central water quality database, and this is freely available to view during using the Google Earth KML files as shown here through this online portal. So this central water quality database provides a fantastic resource that's been extensively studied in South Africa. So next, on to Brazil. So Brazil has several water quality issues. Notably, half the sewage in Brazil is untreated, and there's also pollution from intensive mining and industrial activity. Despite these water quality issues, there's no national water quality monitoring program currently in Brazil. Instead, each Brazilian state has developed their own water quality monitoring regime. And this uncoordinated approach has generated data that's very difficult to analyse at the national level. So Sao Paulo state has become a leader in water quality monitoring in Brazil. It has over 500 sites monitoring up to 60 different parameters bi-monthly throughout the state. And a recent study by Delmedia et al. Analysed the water quality data collected by Sao Paulo state. And they suggest sampling could actually be a lot less frequent and still accurate. But this really depended on the region. So the UGRIH, which is the section the water quality is monitored at, along with the season that's being monitored, and which contaminant is or parameters being looked at in the water. Nevertheless, they found that 2 to 6 samples annually would probably be enough for some of these areas. So thirdly and lastly, I'm going to look at the country of Iceland. And I really picked Iceland because I've just come back from a trip to Iceland, and they take water quality very seriously over there. This is a picture of my daughter at an exhibit on water in Iceland at the Perlan Museum in Reykjavik, and it had loads of information about Icelandic water. For example, where all their national freshwater springs are located, and they had amazing graphics showing how flood peaks can be predicted along major Icelandic rivers. So though Iceland is a very small population and has unique geology, mainly volcanic basalts, it's got fantastic quality drinking water. Over 95% of drinking water requires no treatment in Iceland, and chemical quality compliance is over 99%, which is the highest of any of the 27 EU member states. So this is largely due to Iceland's nationally coordinated water quality monitoring programme, which combines regular and audit monitoring with a programme of measures to make sure that all water bodies in Iceland have at least a good status. This is thoroughly discussed in the Icelandic River Basin Management Plan, which you can see here, and this can be accessed freely online. So just to finish off this section, I want to discuss the GEMS water programme. So one of the slides I used earlier, the background was taken from the Global Environmental Monitoring System for Fresh Water, GEMS Water, who are a UN environmental programme established in 1978. And their goal is to collect worldwide data on water quality. This is so that they can analyse spatial and temporal trends in water quality data, but they also want to encourage a standardised approach to water quality monitoring, which, as you've just seen, doesn't really exist. So all the GEMS water data is freely available through the GEMStat data portal, a link shown here, along with a screenshot of the portal in action, which you'll notice there's lots of data for some parts of the world, notably Europe, and a lack of data in other parts, for example, North Africa. Nevertheless, if you want to access data on water quality and there's not a lot of national data available, this is a fantastic place to start. So now on to a topic I'm a little bit more comfortable with, how water quality is monitored in Scotland. So SEPA currently classify water quality at over 3,000 water bodies throughout Scotland. They're shown here in this map. So you can see they're spread all over the country. They monitor these water bodies on a monthly or even bi-monthly basis, which, as you can imagine, is no small undertaking. As this diagram on the left illustrates, overall surface water quality can be classed as either high, good, medium, poor, or bad. And this depends on both the ecological and chemical status of that water body. So the ecological status combines biological quality elements, for example, water plants and animals, with general chemical and physiochemical factors, for example, oxygen and nutrient levels, and hydromorphological conditions, for example, water flows and fish migration patterns. So to attain a good status, all three of these quality indicators must be good, and the lowest status is taken forward. These statuses are worked out by comparing measured parameters, which I just mentioned, with preset threshold limits. For example, if total chromium exceeded 37 micrograms per litre in the chemical status, then that water body would be failing to achieve good. So F value would be taken forward. And a good status is only achieved if the levels of all priority or dangerous substances are below the EU preset thresholds for these levels. But the threshold limit and which parameters are measured really depends on what the water body is being looked at. For example, if it's a loch, river, estuary, or coastal water body, and also what pressures are affecting the water body. For instance, are there nearby solution sources or known acidification? So overall, SEPA's monitoring programme can involve frequent sampling of thousands of water bodies and measuring up to 78 different parameters. So given this huge monitoring undertaking, SEPA wanted to know if there was a way to improve their current method for monitoring water quality. In particular, they were keen to find out if there were ways to reduce data redundancy while maintaining data accuracy. However, understanding of what data set they had was really limited. So the first step was really to examine their available water quality data. This overall exploratory goal was focused into three more specific questions by SEPA. These focused on the stability and degradation space, if the ecological and chemical parameters used to determine water quality were related, and how this can guide future monitoring trends. So this is what their data looked like. So SEPA kindly provided me with their water quality monitoring data, which is publicly available through their website if anyone's interested. So these data sets are large. The parameter data set alone has over 1 million rows, covering 78 different parameters at over 3,000 water bodies for 15 years. This data set shows the value of the parameter in the result and does each parameter, each water body for each year. The classification data set is a little bit more straightforward as it just gives the overall classification of the water body in question for that year. And there's also information on how certain they are about these results. There's also a whole load of lookup tables that provide a lot of extra information. But as you can imagine, the data required quite a lot of reformatting and cleaning before there was any analysis. So first of all, to examine how a water body varied or fluctuated over time, I used time series analysis plots, and there are examples shown here for different water bodies in the bottom. So the time series plot really compares the water quality over time, so how many years it'd been monitored for each water body in question. I fit a simple linear model to these plots, and the slope of this model indicates if the water body is improving or deteriorating or stable. So the slope values are given in blue here. For each water body, I also calculated the consecutive disparity index, or the D value, which are the green numbers on these plots. And this assesses the degree of difference in subsequent measurements along a time series. So high D values show there's a lot of temporal variability, and D values of 0 indicate temporal stability. So I first calculated the D value for the broadcast water body classified as good or bad for each year. This is because SEPA really wanted to understand where water bodies flipped between good and bad classifications over different years. So therefore, unstable water bodies did change from good to bad, so had a broad D value greater than 0. But I also wanted to look at more detailed classification. So I took 5 different classification values, ranging from bad through moderate up to high. And I worked out the D value for this more detailed classification. This helped me differentiate between stable water bodies and highly stable water bodies. So highly stable water bodies on the far right here have a detailed D value of 0. because basically the water body quality has not changed at all over the time it was measured. But if the general D value was 0, but the detailed D value was greater than 0, the quality class may have changed slightly, but it won't have changed from good to bad. So in this middle example, you can see the water quality has changed from good to high, but it hasn't majorly changed over the time it's been monitored. As I said previously, the water body trend depended on the slope of this best fit line. So water bodies with a slope of 0 were stable. If they were positive, the water body could said to be improving, and if they were negative, then they were deteriorating. So what does this mean with regard to monitoring practices? The temporal stability and trend were combined to generate 5 different groups. So group 1 on the far left here includes highly stable water bodies where the class hasn't changed at all over the time it's been monitored. This is also the largest group. If you look at this pie chart down here, it includes over 1,500 water bodies. And the idea is that these water bodies could be sampled less frequently because they are so highly stable. Groups 2 and 3 are stable water bodies because their class has changed slightly. However, group 2 has a positive trend, it's improving, whereas water bodies in group 3 are deteriorating. So this suggests that, like group 1, group 2 could be monitored less frequently because it's stable and improving, whereas water bodies in group 3 should be watched more closely because they're at risk of dropping down a class. The same is true of groups 4 and 5, which are improving and deteriorating respectively, but these groups are unstable temporally, so it's worth monitoring all of them really closely. Although it's really interesting to note that groups 4 and 5 are by far the smallest groups, and combined they only make up a quarter of all water bodies in Scotland. So I also wanted to look at the spatial stability alongside the temporal stability. And I looked at spatial stability based on the fact that objects closer together in space tend to be more similar than those further apart, Tobler's first law of geography. So I wanted to test if this was true of water body quality and identify where nearby water bodies were highly similar or dissimilar. So I achieved this using Warren's eye, local indicators of spatial autocorrelation, LISA, and also spatial k-means clustering. Now, I'm not going to go into too much detail about this methodology, but I want to show an example for the Tay Basin. So this is the Tay Basin here in the middle of Scotland, which is where I'm based. And I took each basin in turn and looked at the overall Moran's eye, which in this case was significant, so it suggests there is definitely some spatial clustering in this data set. And I looked at the Moran's eye scatter plot shown here. And this was so I could look at local indicators of spatial autocorrelation and identify groups of water bodies where they were all very similar. So these red dots are high, so the water body has a high water, but its neighbouring water bodies also have high water quality. So what I did was I extracted all these spatially significant water bodies for all of Scotland, and I clustered them. K means clustering. So I just picked an arbitrary number for K and looked at how many groups we would get. And as you can see, based on this map on the left, you can see groups of very similar water bodies that will all have high water quality. So therefore, we probably don't need to sample all of these water bodies. We could sample 1 or 2 from each cluster and apply this to the others, which is already being done by an extent from SEPA. They just need to iron out the methodology a bit clearer. So the next question I wanted to address was if the chemical and ecological parameters used to classify water quality were related. Because if they were, there's a lot of redundancy in the measurements, especially if we could predict the value of one parameter from the value of another one. So to answer these questions, I use correlation analysis and principal component analysis. So first of all, correlation analysis aimed to determine how the ecological and chemical parameters were related to each other. I use scatter plot matrices to assess the pairwise relationship between all of the chemical and ecological parameters. I've given an example here which compares macroinvertebrate number of taxa versus the macroinvertebrate score per taxa. And this generated a lot of scatter plots matrices, as you can see here, because it compared all 40 parameters with all other parameters. But what I was really interested in was where there were pairs of parameters with a strong and significant correlation. So basically, if the R-squared value was above 0.5 or below negative 0.5, and the p-value was significant at less than 0.01, which you can kind of see with the dark red and the dark blue in this plot. But to make it clearer, I extracted all 95 pairs of parameters with a strong and significant correlation. And I could only fit the first 25 on this table, but there are lots of really interesting correlations that could be explored in more detail and thought about, but that's a story for another day. So from the 95 pairs of parameters in the full version of this table, of strong correlations with other parameters. And I also worked out the average R-squared value for the correlation between one parameter and all others. All 40 parameters just about fit on this window. You probably won't be able to see it in a whole lot of detail. But what's important was that some parameters have a high number of correlations with other ones. For example, regulatory biological oxygen demand, nickel, ammonium, and phytobenthos diatoms. And this suggests that these highly ranked parameters could be accurately predicted from other parameter values. I also looked at principal component analysis, which is usually used as a dimension reducing tool, but I wanted to use it to see how similar the parameters were. So this is the correlation circle plot, which plots the coordinates of the first two principal components for each of the parameters. And on these plots, parameters which are similar based on all their values will plot close together, and ones that are dissimilar will plot further away. So I used k-means clustering again to group parameters based on their principal component coordinates so that I could find which ones were similar. And overall, there are about 5 groups that come out that you could relate to temperature, the top one. The second one definitely looks at more biological species, for example, dissolved oxygen. The third group focuses a lot on nutrients and wastewater, so biological oxygen demand and pH. The fourth group covers a lot of chemicals you'd likely find in urban or industrial wastewater. And the fifth group covers parameters that you'd find in agricultural runoff or wastewater. So as before, perhaps not all the parameters in each of these groups need to be measured. Perhaps you could measure a key few and then predict the values of some from other parameters. So to test this theory, I used a machine learning algorithm called random forests to predict the value of phytobenthos diatoms, an important ecological indicator of water quality in rivers. So I'm not going to go into detail about how random forests work, but simply they take the value of these input values listed here and look at the relationship between the input values and the output, in this case phytobenthos diatoms. So this is done by building lots and lots of regression trees where a randomly selected subset of the data set, so all the water body measurements, is split at each of these nodes, shown by triangles in this diagram, based on the criteria. So for example, the top split happens based on if that water body has log phosphorus above or below a cutoff value of 4.45. So this tree growing process is repeated 1,000 times, and the trees are combined into a random forest. So one algorithm which can be used to predict diatoms at a water body we don't know about, but we do know what these input variables are, and we can use these to get our diatom values. I also checked the accuracy of my predictions using tenfold cross-validation, which I won't go into detail about unless anyone's really interested. And as random forests are really lots of individual regression trees, like I just showed, there'll be lots of options for our predictions based on a very similar set of input variables. So usually the algorithm just selects the median value, so the 0.5 quartile. And you can compare the predictions for our diatoms based on our random forest at this value, based on and compare them with our actual values. And our R-squared value, when we look at these, is not bad. There's obviously a bit of overestimation going on, but it's 0.43. However, if we look at the prediction for the 75th percentile, 0.75 shown here in the middle bottom, we can see that the agreement between the predicted and actual values is really much closer. The R-squared value is 0.73. So this is called quantile regression forests, and it can be used to determine the likelihood that diatoms will exceed a threshold given a different quantile. And we could also use classification trees to examine how diatoms are likely to be above, high, low, or medium. So this leads nicely on to my final topic about the future of water quality and looking at machine learning in particular. So during this seminar series, we've heard a lot about different emerging technologies that can be used to monitor water quality. From Paul Vreen's talk last week, where he discussed new high-tech imaging and deep learning methods that can be used to quantify plastic pollution, to Simone's talk on how artificial muscle technology can monitor heavy metals. So over the next few slides, I'm going to look at how machine learning methods can be used to model water quality. But to start at the beginning, process-based models are a widely used tool in the environmental sector and have been applied to water bodies worldwide from as early as the 1960s. So in regards to water quality, process-based models use mathematical equations to quantify the biological, chemical, and physical components involved in the cycling of a contaminant through a water body. An example is shown here, a simple 1D vertical lake mixing model for the manganese cycle based on Johnston et al. And some of the common processes that are looked at in the model are listed below. So process-based models are popular and long-lasting for a reason. Their main advantage is that they mimic natural processes using established physical laws and principles, and they're highly accurate when applied to real-world problems. However, they can also be really site-specific, they can be very complicated, they need quite a lot of data to work, and they need a lot of expert knowledge. Therefore, machine learning could be an alternative. So simply, machine learning algorithms work by learning from example, which is what drives natural human intelligence as well. So machine learning models are data-driven. They use statistical methods or data mining algorithms to identify patterns within our input or training data set. And they use this to generate a set of rules or conditions that can be applied to a new data set. This means we can make predictions about something. So in this case, water quality at different locations or at different time periods that we haven't studied. The black box, or the unexplainable nature of machine learning algorithms, is one of their biggest problems. However, visualization can help us overcome this. And machine learning modeling tends to be highly accurate because it can identify complicated nonlinear relationships between input and output parameters in large data sets. So it's perfect for water quality monitoring. I'm going to look at supervised machine learning in particular. So that's just when the training data set has an input and an output value. And there are lots of different types of supervised machine learning algorithms. I don't want to go into it in too much detail, but just to provide a quick overview for those who aren't maybe so familiar with some of these terms. So perhaps the simplest type of machine learning is regression. This builds on the linear regression models that we often fit to our data without even thinking about it. So multiple linear regression just looks at more than one explanatory variable. We can also look at nonlinear regression, where we can fit the best fit curve to minimize our squared residuals, for example, using ordinary least squares. And support vector regression is an expansion of this, where you use hyperplanes to segregate multidimensional space. So we've also got classification methods. Bayesian or probabilistic methods determine the probability that an event or outcome will happen based on the input data. Tree-based methods include the random forests, as I've just discussed. They mirror nature by growing from a root or input data and split into branches and leaves to make some predictions. And neural networks are modeled on the human brain, and they attempt to match some output nodes with input nodes by iteratively adjusting the weights between some hidden layers. So to look at a few examples of where machine learning's been used to predict cyanobacteria in particular, this is because cyanobacteria is a much studied topic in water quality because there's so much data from it. Therefore, there are hundreds of different papers and studies that have used machine learning to predict cyanobacteria and their byproducts. And I'm just going to look at 3 different ones in particular. So first of all, Francie et al. Developed a real-time model that could predict the likelihood of microcystin concentrations. So that's a cyanotoxin produced by harmful algal blooms. And they wanted to know when the microcystin concentrations would exceed recommended guideline values at 8 sites in Ohio, including Lake Erie, as shown here. So they used multiple linear regression to estimate this exceedance probability. And they input environmental data, which include stream flow data along with atmospheric data, so rain levels and air temperature, seasonal data, so the day of the year. And they included this with probe data. So this provides continuous real-time measurements on the water pH, the microcochian fluorescence, which can help us determine what the cyanobacteria is going to be like. As these are lagged variables, they were able to provide an early warning system, so up to 2 weeks ahead, for the microcystin exceedance likelihood, and they had a 90% sensitivity value. So this kind of shows that although linear regression can not be the most complex types of machine learning, if the right type of data is input, it can generate highly accurate short-term predictions. So my second example looks at more long-term forecasting of cyanobacteria, which is usually achieved using process-based models like ProTech. However, Yang et al. Combined 3 different types of machine learning methods: support vector regression, principal component analysis, and backpropagation artificial neural networks, shown here. They input 11 years of historical monitoring data. And this large data set and hybrid methodology is shown in more detail in this diagram, and was used to estimate long-term spatial patterns in lake surface water temperature for a lake in southern China. And these water temperature predictions were then fed into water quality predictions, which, for example, looked at the likelihood of cyanobacterial growth. So this allows us to understand future changes such as climate change and how this could impact water quality. So there's also been a trend of hybrid or combined modelling. And this is basically just when several different modelling methods are put into one, as we just saw for the lake surface water example. And this hybrid methods usually improve the water's overall predictive accuracy and adaptability, but they can be very complicated and difficult to understand. But a really good example of combined modelling using a process-based model and a machine learning model was done by Hansen et al. So they developed a process-guided machine learning model, specifically process-guided recurrent neural networks, to estimate phosphorus levels for a lake in Wisconsin in the USA. So they firstly designed a process-based model called Process, which had a lot of inputs. I've listed some of them here. If you look at the paper, there are a lot more. But this model was able to really accurately predict phosphorus concentrations for the next year or so at the lake. And then they took these phosphorus concentrations at different parts of the lake and in the sediment and put them in a process-guided recurrent neural network to help understand more about the predictions. And they also made sure that the process-guided neural network had ecological constraints. For example, they ensured that phosphorus from one day to the next change wasn't going to be too big. And they also made sure that the predictions up and down the lake weren't a big jump with depth. So this model was overall trained using 20 years of data and validated using cross-validation. And the authors attest that the benefits of process-guided machine learning include high accuracy and reduction of bias, although it does require a lot of data. So lastly, how does this all relate to water quality monitoring? So first of all, we clearly need high quality data to make any predictions about water quality, regardless of our spatial or temporal scale. This will involve collecting high quality data for a lot of water quality parameters, including hydrological, climatic, anthropogenic, and biological parameters, which I've listed here. These can include things like turbidity and pH and dissolved oxygen, and they can be determined using physical samples and analysis. But we could make use of new technologies, including probes on buoys or in situ, and also remote sensing technologies. So climate data is often included in the models, and it includes parameters like air temperature, wind, and precipitation. And climatic data is fantastic because it's usually freely available and predicted through national meteorologic agencies. Some countries are lucky enough to have a wealth of accurate water quality data spanning decades, whereas the data coverage is a lot more patchy in other parts of the world. Local conditions mean that when a model is trained, it tends to be more accurate for that condition, for the local data, for example, for a single reservoir or a river catchment. However, we want the models to be transferable and not overfit to that area. We want them to be applicable to a much wider regional scale. We also need to consider our temporal scale. Do we want to know what the water quality is going to be a couple of days ahead? Or do we want to be able to understand long-term changes that could be driven by climatic and anthropogenic forces? To understand all these different inputs and factors, decision support systems are a really good way of managing these. And this means that water managers can make informed decisions that affect water quality. So generally, water quality parameters are modeled using process-based models. So these are the circular examples shown at the bottom here. But machine learning methods are gaining popularity, and I've listed some of the main ones with these rectangles. Please note this diagram only shows recommended modeling methods and collection techniques for different scales, but they are applicable to a wider range. I was just looking at where they're usually used. So process-based methods are traditional as they replicate natural processes, and they can be accurate if we've got the right data. However, machine learning methods have become popular with the increase in computing performance, and they've accurately predicted a range of water quality parameters at varying spatial and temporal scales. They also need lots of data and need to be carefully operated. And most of these models are open source and can be run using a whole range of programs, including R, Python, GIS, and graphical user interfaces. And as I said before, combined modeling has become very popular in water quality prediction because it tends to increase the accuracy of the models, but we have to be careful about this increased complexity. So I realize I'm running out of time, but the key take-home messages from this presentation are that water quality monitoring aims to ensure that water resources are fit for purpose. So this primarily involves making sure they don't cause human, environmental, or economic damage. To make sure this is the case, there needs to be frequent sampling and measuring of the physical, chemical, and biological properties of a water body. And water quality monitoring is run by governing bodies in countries all around the world, but it's typically costly and requires a lot of time. So analysis of SEPA's water dataset in Scotland indicates there is redundancy in their current monitoring programme, and machine learning modelling could offer a viable alternative. Machine learning models have been used in other locations as a means of predicting quality parameters over different temporal and spatial scales, ranging from short term, a couple of days, to the long term, hundreds of years into the future, and for different spatial scales, from an individual water body right up to a whole region. So machine learning models will only be accurate if the high quality data is input, and this requires long-term data of a lot of water quality parameters, including hydrological, climatic, biological, and anthropogenic data. Collecting this high-resolution spatial and temporal data on these parameters can be challenging, so high-tech options are being used, for example, probes and remote sensing. And there are lots of modelling options available to predict water quality parameters. from the traditional process-based models to recent machine learning models, and a combination of the two. There's no one correct model. It really depends on the spatial and temporal scale of the prediction, along with what data is available. Nevertheless, this is an exciting time for new modelling options to predict water quality, and this could be of great benefit to the wider society. So thank you so much for listening. Please let me know if you've got any questions.
Thank you very much, Sarah. That was a really great coverage with wide-ranging examples of different water monitoring approaches, and also thanks for exploring some of these machine learning models options. I think there's a lot, certainly for me, to learn going forward from here. Do we have any questions for Sarah? Oh, I see some hands up already, so I don't know who was first. Craig is the top one on my screen at the moment, and then after that it'll be Julian.
Thanks. I think Julian was first, but since you've chosen me, I'm going to go first. Sorry, Julian. Sarah, that's super interesting. I guess my question is, we've also had a project where we're looking at using machine learning, and I agree it's very important for the learning part that you get high quality data to teach your machine how it responds to the data. Is it necessary to have such high quality data afterwards, once the machine is already set up and running? And I mean, I guess I'm thinking, you know, you might use cheaper pH probes or something in the environment, which would not be as accurate necessarily, but can the machine not learn that as well and give you like different ranges, I guess, or different levels of accuracy? Maybe I'm trying to think my question out loud, which is probably less helpful for you.
No, I think I get where you're going at, and I think it's a really good question. And it's what I think Paul talked a little bit about last week. What quality of data do we need to monitor plastics accurately? I think there are methods where you can put the measurement accuracy in and make sure it's within the range, but I'd have to investigate that in more detail. But I do get your question, and I think it's definitely worth thinking about in the future, if that makes sense.
Yes, absolutely.
Julian?
Thanks very much, and hi. First of all, that was a very nice presentation. You covered a lot of, I think, complex stuff, but made it very accessible. And I think I have a question, but I have a general comment first. And I think I would agree that machine learning approaches, I think, are a natural progression from the process-based models as we've started to increase these data sets over decades. So with that data, it makes sense to use the data to improve our understanding or predictions or modeling. I disagreed a little bit in that I think process-based models, in a sense, are more universal, and the machine learning ones are trained on a set of data, so they work for that set of data. So there may be more site-specific. Theoretically, at least, they're not as portable, maybe. And I think it's worth noting that, of course, in many parts of the world, the data isn't there. So in some way we need to develop those process models further, so they're maybe more widely applicable. Anyway, here's my comment. In a lot of this machine learning modeling, you use a lot of data and you have to transform this data. And so sometimes it's the transformation of data which is the key step to producing a good model. And since you come from a geochemical background, I was assuming somewhere you might talk about compositional data and whether compositional data and transformations associated with that are something that we should take into account with water quality modeling and perhaps machine learning modeling.
Yeah, I think that's a really fair question. I mean, it was actually the question I was asked in my PhD Viva about compositional data, because it applies in all geochemical data. And you're right, transforming it can change the ratios. And I'm not sure, I've not read a lot about compositional data in machine learning. So I think that would be something to explore in a lot more detail. And I think your comments about process-based modeling is valid. I think, yeah, they do have a lot of scope still and are well developed. But I just wanted to cover kind of their interaction a little bit with machine learning as well. But thank you, your points were really interesting.
Thanks very much. So maybe let me know if you ever explore compositional data and machine learning.
Yeah, I'd like to. Yeah, we can talk about it at some point.
Sarah, I just wondered how able those who are responsible for regulating our waters, like in Scotland, it's obviously Scottish Environment Protection Agency, but how much of their capacity is just tied up in meeting current regulations, and what is their capacity to embrace some of these new technologies?
Yeah, I think very good point. So yeah, a lot of current research is just meeting the regulatory demands, and I'd like to know if it was different in other countries. I don't know, as I say, a whole lot about how it's monitored. But yeah, definitely in Scotland, a lot of their focus is on meeting the guidelines and maintaining the water quality, and they don't think too much into the future, which is kind of what I was hoping to show them, and to show generally with this machine learning and new technologies, they can offer big cost savings in water quality monitoring, and they can still be accurate. We just need to set them up correctly.
Yeah, and they may also be curtailed by the way that they make the measurements and the way they have to account for
Yes.
their quality control as well, I guess.
Yeah, no, definitely. I think quality control is very important, and checking of all of it. And yeah, meeting the guidelines is essential.
See if there's any more hands. Oh, there's another hand, yes.
Good afternoon.
Hi.
Yes, I just wanted to find out in there's a slide where you showed the categorization of water quality in terms of those water bodies that have got high quality, good, moderate, and so on and so forth. What variables did you use to do that? And did you try maybe to use categories of variables in terms of those that are heavy metals, those that are organics, and those that are perhaps toxics, and so on? And what weighting did you give to those, and how did you arrive at those categories? And then secondly, I saw in your other slide where you were showing the trends, temporal stability and trends, where you were showing data for a particular year. It would seem that it has been averaged for a particular year. How is the seasonality taken into account in that? Because some years you'll have more rain, which actually dilutes the quality and so on. So how is that taken into account? Thank you.
Yeah, those are two great questions. So second one first, because it's in my mind. So yeah, I think I would really like to understand more about the seasonality impacts on some of the parameters. Unfortunately, the data set that SEPA provided was just an average annual measurement for each of the parameters, but I would really like to get more in detailed kind of temporal measurements throughout the year and see how these vary year on year. Different summers, as you said, if they're wetter, drier, does that cause a different concentration of some of the parameters? And I think the first question asked about the categorization. Was that the principal component or just the general categorization? You can clarify a little bit more, that'd be helpful.
Well, it's the one that I saw in your presentation where you're showing the high model. I think the other one was medium, bad quality, good quality, and so on.
Okay, yep, I'm with you. Yeah, so that's how SEPA classify theirs, and they don't weight them. My understanding is they just take the worst quality forward. So if it fails on one, it fails on all. And if it's got a bad quality for the chemical status, that's taken forward and combined into the following one. But yeah, weighting based on other things would be an alternative way of doing it. It's just how SEPA's algorithm is currently set up.
And that's restricted because of the European framework directive.
Yep,
so that they have
correct.
to adhere to that. And it's complicated, but that's
Yes.
the way it's done currently. Julian. The hand went down. Ah, right.
Yes.
Okay. Yeah.
Thanks.
Okay.
Got it now.
That's fine.
Like,
So I think we've
years,
actually
years, and still forgetting to unmute myself. Yeah, just a general comment in that it did seem to me that SEPA is still following the Water Quality Framework Directive, which
Yep.
I was surprised at, but it's good to see that. But I guess my comment, Sarah, was that you did some nice analysis with showing that, of course, you may be able to reduce monitoring or sampling for certain parameters because there's a strong correlation between them. But I did wonder how much of that correlation was, within the same methods, and how much real savings could you get? So, for example, there might be a strong correlation between different trace elements, which are all monitored by or all measured by ICP-MS. So if you put the sample on,
Yep.
you get them out whether you want them or not. And the same, I think there was a correlation between different polyaromatic hydrocarbons. So they're all going to come out anyway. And maybe it's correlation within the analysis that are very strong because they all use the same technique. So I did wonder.
No, I think
Through
that's
all that correlation,
yep.
how much savings or how much could really be thrown out? Like, could you measure a trace element and never need to monitor PAHs?
Yeah, no, I think that's a really, really good question. And it's kind of where I wanted to go next. If I did work with SEPA in the future, I want them to kind of give a list or a breakdown about how expensive and how difficult it is to monitor. So I think nickel came out as a really kind of high correlated factor, but there's not a lot of point in predicting nickel because you're right, if you're doing ICP, you can just analyze them all together, which is why I was kind of focusing more on some of the biological indicators that maybe are a little bit more complicated for them to quantify, but it's something I'd really like to look at in more detail in the future.
Yeah, thanks. That's a good point. And that, of course, it's easy to measure maybe lots of trace elements, but if they help predict the outcome of a biological one, which is a kick and sweep and time consuming and counting, then it's a good approach. Thanks.
Thanks.
And then just one more question, I think, Rachel.
Hi, yes, thanks. It's Rochelle from Future Water UCT. I had a sort of follow-on question in a similar vein in terms of you've got this amazing database of so much information, and then you use your machine learning to reduce the amount of monitoring that you do. But then in the future, that would reduce the accuracy of future machine learning models because you're then collecting less data. So is that not also something one should bear in mind before cutting the monitoring programs down?
Yeah, no, I think that's a really valid point because obviously I think we talked about quality assurance. You need to check the data with some monitoring, but you're right, the whole idea is to reduce the monitoring, which means that, yeah, there'll be less in the future to work with. So I think that's a very good point.
Yeah, I agree. And I think SEPA were looking for the ways that they could save some resource and maybe target it so that they would do the full suite of determinants only at places where they really need to do so because the water quality is deteriorating. So I think they were looking for some guidance in that respect. So areas where we might not need to make as much because they're very, very stable over very long time periods, but then those that are maybe more subject to change or more affected by current climate and other issues that we face. So I think that's where they were coming from. But I think at this point we should stop because it's 2:04. And I would like to thank Sarah not only for her presentation today, but also for her role in organising this excellent lecture series. Thank you also to all 11 presenters who have given us their insights into current and future water-related challenges from around the world. So just to remind you, all of these lectures are available on the IIS YouTube channel. And there are also, I think there's a course on isotope hydrology coming up in April if people are interested in registering for that. You might also want to look at the past catalogue, and there's you could delve into the lecture series on mangroves. There's one on microplastics, and there's also one on water pollution mitigation and aquatic remediation, among the others. So I think I'll just pass over to Craig now, because I think he would like just to say a final few words before we end this last session.
Thanks. Okay, so I don't have a camera anymore. It chose not to allow me. It's better than showing everyone my legs, which it's been doing a lot recently. Yes, so I'd like to say thank you to Sarah and to Margaret and to everyone who's been involved with this series, and also especially to Raquel at Trent University for all of their assistance in making this work. It's really been interesting for me to work and to listen to all of you give different perspectives from different parts of the world, and also to sort of get to know a little bit about who all of you are. And then just a reminder, I guess we should also acknowledge all of our funders for this series, which are Europa, who've funded the program, as well as the Claude Leon Foundation, who fund my chair, and also the IIES, which is the International Institute for Environmental Studies. And then to remind you all that we are having a conference soon. If you don't know about it, that's at Wits University on the 6th and the 7th of April. And there is a graduate... Sorry, it's a long day, Margaret. I'm running out of words. It's a graduate something forum, graduate workshop something.
Graduate forum.
Thank you. Graduate forum at the Wits Rural Facility, which is straight after that. So if you're part of these organizations, please have a look at these things and join us, and try and participate if you can as well, because I think it's really focused specifically on younger researchers, and we are trying to really focus at master's and at PhD level, and in fact honors level as well, if you're out there, just to give you a friendly platform upon which you can present your work and also share what you know and learn from others across the world. So yeah, thank you very much. And like Margaret said, an especially big thank you to Sarah for your work on the series. I think without you, it would have been a real flop. So am I allowed to say that? I think so. I'm going to say that because I think it's true.
Thank you, Craig.
Yeah, thank you, Sarah.
No, thank you everyone for coming along.
Cool.
Okay, so thank you very much, and hope to meet some of you soon.
Brilliant. Thanks, everyone.
Bye bye.
Cheers.
Okay.